Welcome to this briefcast on DNA fingerprinting. DNA profiling or DNA fingerprinting has revolutionized the way forensic medicine is practiced in this country by tapping a relatively unpretentious region of the human genome. Scattered throughout our chromatin are regions of repetitious non-coding DNA of varying length where a core sequence of nucleotides is repeated in tandem, that is, in a head-to-tail manner over a stretch of DNA fiber. Repetitive DNA, which accounts for more than 50% of the genome, is DNA for which no function has been identified and is often referred to as junk DNA. Some repetitive regions of DNA are designated microsatellite DNA or STRs for short tandem repeats, when the repetitive core sequence is only a few nucleotides long at one hypervariable location or locus. Other regions are called mini-satellite DNA, when the core sequence is on average 35 nucleotides long and it occurs at many locations or loci in the nuclear genome as variable number tandem repeats. In a criminal investigation or in a paternity dispute, DNA fingerprinting would entail the isolation of total DNA from hair, semen, epithelial cells, or blood from an individual of interest and from blood or hair sampled at the scene of the crime. The DNA samples are digested in vitro with a restriction enzyme that does not cleave DNA within the core sequence. Digestion of DNA produces a collection of DNA fragments of different sizes, some of which contain tandemly repeated core sequences, while others do not. Agarose gel electrophoresis is then used to arrange the fragments according to size. Following in-gel denaturation of DNA fragments and blotting of the gel contents onto medium, such as nitrocellulose or nylon membranes, the blot containing immobilized single-stranded DNA fragments is blocked and probed with a radioactive single-stranded oligonucleotide designed to hybridize with core sequence. After an incubation period, immobilized DNA fragments containing repetitive core sequences that hybridize with the radioactive oligonucleotide probe are detected on X-ray film as dark bands by autoradiography. Since the core sequence is tandemly repeated in variable numbers at multiple locations or loci throughout the DNA, the autoradiograph shows a ladder of dark bands at each lane that resembles the barcode that stores use to identify merchandise. The genetic barcode is unique to each individual. Half of the fragments in the DNA ladder are inherited from one parent, and the other half corresponds to fragments inherited from the other parent. The forensic scientist declares a match when two bands on the autoradiograph, a DNA fragment from the suspect and a DNA fragment from the biological samples collected at the crime scene, unambiguously have the same mobility and therefore migrate at the same position on the autoradiograph. In a ladder of DNA fragments, the slow-moving fragments in the top half of the autoradiograph are easier to score because the fragments are farther apart than the fragments in the lower third of the autoradiograph, where bands tend to migrate in close proximity. If the probability that two unrelated individuals share a band is 25%, the probability that two individuals share multiple bands will be 0.25 to the power of n, where n is the total number of clearly visible matching bands. In a profile with 25 distinct matching bands, the probability that the accused is linked to the evidence gathered at the crime scene is 0.25 to the 25th, or 1 in more than a quadrillion, or 10 to the 15th. As of 2008, the world population is only 6.7 billion. 13 microsatellite loci or STRs for short tandem repeats on 13 non-homologous chromosomes plus an STR locus on the X chromosome and a locus on the Y chromosome are used for DNA profiling by the FBI. For example, the TPOX locus, which is present in human chromosome 2, has 9 alleles designated 6 through 14 according to the number of copies of the repetitive element AATG. This means that allele 12 at the TPOX locus has 12 copies of AATG repeated in tandem, while allele 8 has only 8 copies of the repetitive AATG sequence. The D16S539 locus, which is present in human chromosome 16, has 9 alleles designated 5 through 15 according to the number of copies of the repetitive element GATA. Since there are two homologs for each chromosome, each individual has two alleles at an STR locus. A homozygous individual at the TPOX locus has either two copies of the same short tandem repeat, for example, one allele 8 inherited from each parent, whereas a heterozygote would have two different alleles, for example, an allele 8 inherited from one parent and an allele 12 from the other parent, or an allele 7 from one parent and a 10 from the other parent. The polymerase chain reaction, or PCR, is used to identify the hypervariable alleles at the 13 STR locations identified by the FBI's CODIS for Combined DNA Indexing System, because PCR amplification for STR sequence analysis is faster, convenient, and highly reliable. The analysis of STRs begins with sample collection. In the laboratory, the DNA is isolated and purified, and locus-specific forward and reverse primers annealed to unique DNA sequences located just outside the STR sequence are used to amplify the DNA sequence at specific STR locations. After amplification, the PCR products are separated according to size by gel electrophoresis alongside a ladder of markers with all possible alleles at the STR. Since the PCR products are fluorescently tagged as they are synthesized, the size of a PCR band on the gel can be determined and its corresponding allele numerical designation can be established from the ladder of allele markers at its side. Every lane on the gel to the left has one or two PCR amplified fragments instead of the complex ladder of DNA fragments obtained with a multi-locus probe as illustrated on the gel to the right. As you can appreciate, PCR amplified bands are easier to score on the gel to the left than on the gel to the right. The frequency of all hypervariable alleles at any given STR locus can be obtained from population databases. For example, at the D16S539 locus, the frequency of allele 8 among Caucasians is 0.025, it is 0.1 for allele 9, 0.07 for allele 10, 0.27 for allele 11, 0.37 for allele 12, 0.16 for allele 13, and 0.03 for allele 14. Therefore, the genotype frequency for a homozygous Caucasian carrying two copies of allele 13 at the D16S539 locus is obtained by multiplying the allele frequency 0.16 times 0.16 or 0.0256 or about 2.5%. On the other hand, the genotype frequency for a heterozygous Caucasian with one copy of allele 12 and one copy of allele 11 is obtained by multiplying 2 times the frequency for both alleles, in other words, 2 times 0.27 times 0.37 or 0.199 or about 20%. The profile frequency, which takes into account the allele frequencies at all 13 STR locations, is obtained by multiplying the genotypic frequencies for all 13 STR locations. The example presented on the slide shown here calculates the profile frequency for only 4 of 13 STR locations: CSF1PO, TPOX, TH01, and VWA. As you can observe on the table, the individual under consideration carries allele 10 and 11 at the CSF1PO location. The homozygous for allele 8 at the TPOX location carries allele 6 and 7 at the TH01 STR and is homozygous for allele 16 at the VWA locus. The genotype frequency at the CSF1PO site is the product of the allele frequencies 0.25 and 0.31 times 2 or 0.16. The genotype frequency at the TPOX locus is obtained by taking the allele frequency of allele 8, 0.53, and raising it to the power of 2 to yield 0.28. Similar calculations are used to determine the genotype frequencies at the remaining 2 loci, TH01 with a calculated genotype frequency of 0.07 and VWA with a genotype frequency of 0.05. The profile frequency, which is the product of the genotype frequencies at the 4 locations, or 0.16 times 0.28 times 0.07 times 0.05 is 0.00014. This means that the probability of finding 2 Caucasians sharing the same alleles at all 4 STRs is 1 in 7,142. Although the probability of finding Caucasians with identical genotypes at the 4 STR loci we have just discussed is very high, incorporating genotype frequencies calculated for other STRs should allow us to establish whether or not the presumed father is the biological father of a child in a paternity dispute, or whether or not the suspect can be linked to the crime scene. In a paternity dispute or in linking a suspect to a crime scene, it is necessary to analyze at least 9 or 10 STR loci in order to arrive at an unambiguous conclusion. This concludes this briefcast on DNA fingerprinting. Thank you for your attention. Live strong.